Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's webinar is on vaccine and biotherapeutic formulation development and manufacture using spray drying. My name is Rebecca, and I will be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and following today's presentation, there will be a Q&A session. This webinar is designed to be interactive, so please feel free to submit questions and comments using the questions chat box. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance at all, please contact me at any time by sending a message using the same chat panel. At this time, all participants are in listen-only mode, and the presentation slides will advance automatically for you today. Please note that this event will be recorded and made available for future download. At this point, I would like to thank Bend Research, who has helped develop the content for today's presentation. Bend Research is a leading independent scientific development and manufacturing company, and for more than 35 years, the company's multidisciplinary teams have pioneered and advanced technologies to solve their clients' most challenging technical problems. The company's capabilities include drug delivery technologies such as spray drying, hot melt extrusion, and controlled release, as well as inhalation and biotherapeutics development. Spray drying can be applied to a number of key biotherapeutics, including peptides, proteins, and virus-like particles for pulmonary delivery, RNA, DNA, and proteins for parenteral delivery. Bend Research offers process development and cGMP manufacturing capabilities to produce spray-dried dispersions. Its spray-dried methodology minimizes the use of valuable active pharmaceutical ingredient, shortens process development timelines, and ensures scalable process and particles. Bend Research employs nearly 300 people and has 6 state-of-the-art facilities located in Bend, Oregon. I would now like to introduce our speakers for today's event. Dr. Jeff Bright, who is Director of Inhalation Technology at Bend Research, will be speaking. Dr. Bright is responsible for inhalation drug delivery, biotherapeutic development, and formulation technologies. He previously held the position of project leader and research scientist at Bend Research. Dr. Bright earned his PhD in pharmacology from the University of South Alabama while working at the Center for Lung Biology. His doctoral work focused on gene regulation involved in pulmonary hypertension, and Dr. Bright performed his postdoctoral work at Roche Pharmaceuticals in Palo Alto, California, where he focused on the genetics of complex disease states. Dr. Bright has been with Bend Research since 2007. Joining Dr. Bright today is Mr. Devin Dubose, who is Senior Research Engineer at Bend Research. Mr. Dubose's responsibilities include spray drying development and scale-up for preclinical and clinical projects and transferring spray drying processes into a cGMP environment. He also has expertise with special spray drying projects for biologics and inhalation. Mr. Dubose has been with Bend Research since 2007, and he graduated with a Bachelor of Science in Chemical Engineering from Oregon State University and is a registered professional engineer in the state of Oregon. Mr. Dubose has 3 U.S. patents pending currently. So without further ado, I'd like to hand over this presentation to our speakers today, and they may begin when ready.
Thanks, Rebecca. So this is Jeff, and today I'll be talking about a vaccine and biotherapeutic formulation development and manufacturing using spray drying. First of all, we'll talk through a quick intro of Bend Research. Bend Research is an independent scientific development manufacturing company. Our teams of engineers and scientists advance clients' technologies to solve problems in the pharmaceuticals and outside pharmaceuticals as well. Our mission is to improve the standard of living through development and application of technologies and our expertises that we develop in-house, internally at Bend Research. We have a sort of laundry list of what we call our sort of Boy Scout values of curiosity, science and engineering excellence, quality culture, reliability, focus, integrity, honesty, family, loyalty to customer, reputation, and our Bend Research mantra of no whining. which means essentially get to work and get the job done. On the right-hand side of this slide, you can see our bricks and mortar, where we actually work every day, and we have our R&D facilities are shown in the upper right-hand corner here of this slide. We have R&D labs. We also have an engineering and pilot plant facility. We actually perform spray drying. That's where our stainless steel lives. We do technology transfer from our R&D facility from the glass to the stainless steel. It's perfected by our spreader engineers. Powders are manufactured. And then, as Rebecca was saying earlier, Devin and the engineers will actually transfer these processes from the pilot plant into our GMP manufacturing facility. And we actually do have a new higher containment facility that meets low bioburden specs, which actually specializes in high-potency compounds and inhalation compounds as well. And also for biotherapeutics, this is critical importance. We're planning a future expansion, 2015, of commercial spray drying capabilities, which is currently in development. And please stay tuned about those sort of notifications in the future. I think we're not advanced. Sorry, we're having technical difficulties. There we go. Thank you. So, technology overview. Bend Research is really known for solubility enhancement and spray dry dispersions. So the left-hand side of the slide, we actually have our sort of oral capabilities, which focuses on SDDs for hot melt extrusions. We also perform solid nanocrystalline dispersions, nanoparticulates. Various technologies are currently available in-house to actually enhance solubility of low bioavailability compounds. In the bottom left-hand corner, we see our modified release or oral cell dosage forms capabilities. We have immediate release IR tablet capabilities, monolithic tablet capabilities, multiparticulates, melt spray congeal. These are some of our offerings we have in-house. On the right-hand side of this slide, we also have our sort of more designer particulates and biotherapeutic sort of applied technologies. Upper right-hand corner, we have our inhalation and particle engineering capabilities, including our GMP facility, which is shown in the pictures. This is sort of an important area for biotherapeutics as well as small molecules. In the bottom right-hand corner of the slide, we see our bioprocessing and biotherapeutics capabilities. We're currently expanding into the production and understanding of manufacturing biotherapeutics. We're also working in areas, this webinar is focused on bioformulations as well, which is sort of a soup to nuts approach of biotherapeutics from manufacturing, all characterization to formulation and characterization of those formulations as well. So we've sort of got our feet wet back in the day in small molecules. We're now expanding and applying technologies and know-how to biotherapeutics currently. So a rough outline of this webinar, what we'll be talking about today between Devin and myself. We'll be talking about why someone may or may not want to spray dry biopharmaceutical. Historically, lyophilization has been the key technology to sort of create a solid dry powder or a dry cake formulation of biopharmaceutical. Recently, we've been asked by clients to look into spray drying to solve problems in the solid state for formulation of biotherapeutics. We'll be discussing why that might be applicable or might not. We'll also be doing a deeper dive into spray drying itself. That is how the process defines the product. Devin will be doing some very neat talk, have a very neat discussion around engineering spray drying itself and biotherapeutics and the handles he and engineers sort of pull on to develop a quality product. And dovetailing off of that, and actually along with that, we'll be discussing a high-level approach of using formulation to solve stability issues. We'll be discussing why or why you may or may not want to include excipients in a formulation. We'll also then discuss a roadmap, formulation and process roadmap to actually describe and be quantitative about defining a system or methodology of creating and developing and pushing forward into GMP a spray-dried biopharmaceutical formulation. And to top all this off and sort of tie it all together, we'll have 3 case studies that we'll be discussing. We're doing sort of a deeper dive there and how clients have approached us with certain questions and how we solve those those problems for our clients using spray drying and formulation applications. To get us all on the same page, here's a rough definition of a biotherapeutic. And so historically, a biotherapeutic has been referred to as a material that's been produced using biological means. And some of those sort of compounds are shown in the bottom left-hand list there of the slide. These sort of compounds might include monoclonal antibodies or mAbs, vaccines. We're discussing today virus-like particles, both enveloped and we worked with enveloped and non-enveloped VLPs. We worked with designer proteins. These are produced biologically, but through recombinant DNA technology. We've also worked with subunit vaccines, synthetic peptides, antisense oligonucleotides, as well as siRNA. The last three bullet points there are not necessarily made biologically, but are made synthetically. But for this conversation, because their subunits are biologic subunits, quotes, we will actually lump those into biotherapeutics here for this conversation. So in this slide, sort of a introduction to our case studies and some problem statements we've seen come across our bow here at Bend Research. We are a contract-based company. There's internal R&D efforts occurring here at Bend Research, but predominantly we do try to solve problems that people bring to us to get a better feel for what the industry is doing and where it's heading. And these are some of the reasons people have actually come to us and asked us to spray, dry, and formulate biotherapeutics. The first one, this first bullet point, the top being elimination of cold chain storage. This is applicable for developing nations where freezers aren't readily accessible. As can also be applied to supply chain management here in the United States or in Europe for transporting material that might not need to be frozen, but theoretically could be spray dried, transported dry, and then resuspended when needed. And this is sort of an alternative to lyophilization, which is the second bullet point. And spray drying itself is a continuous, robust, and scalable process, as Devin will dive into a little bit here in a few minutes. We have perfected a sort of process train that mitigates or minimizes the need for API during formulation development. We're discussing that briefly, which people have asked us to look into quite heavily. As we can imagine, these biopharmaceuticals are fairly costly. People often consider them very precious. We brought these compounds in-house and built a process trained formulation on using an API-sparing process. And along with that process, we've also developed a formulation approach that looks at excipient compatibility with the process, with the API itself. And these approaches, these first two bullet points, we'll actually be highlighting in our first case study, which was a collaboration with PATH, looking at a vaccine formulation that we wanted to develop that would have 6 months stability at 50 degrees Celsius. Back to the left-hand side of the slide, people have also asked us to look into high concentration reconstitution of biologics such as monoclonal antibodies. The goal here being we would spray dry at a fixed concentration of monoclonal antibody, create a powder, and then have essentially the ability of our client to resuspend at a given desired concentration, theoretically at a higher concentration than they started off with, to mitigate sort of flocculation or aggregation events routinely observed in process ops such as UFDF, TFF, things of that nature. We'll be discussing that sort of approach in our second case study, which is a mAb formulation. They wanted to see room temperature, solid state stability for downstream high concentration reconstitution. We'll be discussing some formulation approaches from that case study, as well as some engineering approaches, as well as some analytics that went along and with that program and justified our approaches. The last bullet point here is titled particle engineering. Some of our clients have come in and asked us to formulate biotherapeutics for targeted delivery, in this case either pulmonary or intranasal delivery. And what's important about that is particle design itself, stabilization of the API. These are small particles that and also have to be aerosolizable. And what's important there is also flowability, filling, and reconstitution, as you can imagine. Rate has to be fairly quick, and that all ties back to formulation and product morphology. We'll be highlighting some of these topics in case study number 3, which Devin will be discussing, which is a look at engineered particles, where we actually formulated and manufactured a norovirus, virus-like particle formulation for effective intranasal delivery as a dry powder formulation. So these are the general concepts we'll be touching on during the course of this talk, along with analytics and engineering overlaid on top of those concepts. And we'll be wrapping this discussion up with a discussion of these case studies at the end of the webinar. At this point, Devin will be jumping into a deeper talk around the engineering of biotherapeutics and spray drying, and discuss some of the handles and hurdles, I guess, to creating these formulations and what they've done internally to mitigate and actually some of these concerns and actually turn them into advantages here for formulation and process.
Thanks, Jeff. So slide 7 just provides an overview of why the spray drying process is applicable to these biotherapeutic formulations. Thermally, it's a relatively gentle process. So due to evaporative cooling during the drying or solidification part of the process, the material or droplet and ensuing particles kind of shielded from some of the more extreme temperatures experienced at the inlet temp, such that really the maximum exposure is more akin to the mild temperatures experienced in the outlet of the spray dryer. Likewise, there's kind of multiple tuning handles that surround the process to optimize things like drying kinetics in the solidification process, atomization and the desired particle size or the desired water contents for storage and long-term stability for a given product need.
And I'll jump in right there real fast, Devin, that's okay. So Devin mentioned water content. That's critically important for a biotherapeutic to actually regulate and understand water content and how that impacts API integrity. And so Devin actually touching on that as far as water content control is an important part of this process.
Thanks, Jeff. The last bullet there is just that this is a highly scalable process and proven commercial viability, which takes us to the next slide, slide 8, which highlights kind of the bin research scale or spray drying process train. And the things to highlight here is within this process train, we can manufacture sub-gram quantities all the way up to hundreds of kilo type quantities. Similarly, a lot of these dryers on the left are just designed and built by bin research to kind of optimize the material usage at the early development phase and really be material sparing operations, but still highly flexible and scalable into the larger scale spray dryers. Slide 9 is designed to try to link kind of the critical aspects of the formulation to the process, which are, it's highly critical to understand the different spots in the spray drying individual unit operations that can destabilize the formulation, and to also understand how to design the formulation to work around any of these potential failure mechanisms. So just as a simple example, we've seen issues with solution tank materials of constructions of actually causing flocculation or aggregation of different formulations, and used equipment design constraints or formulation fixes to get around issues like this. Other stresses, the shear stresses at the atomizer, forming the actual droplet, or thermal stresses in the actual drying process can be other spots where the formulation could be denatured. And all this kind of links back to understanding the key physical situation and what kind of materials or excipients might be needed for the desired problem statement, whether for long-term stability or desired water content, and what that means for some of the packaging and storage requirements, which Jeff will jump into later in the presentation. Slide 10 takes that same kind of concept but overlays the actual physical situation that's happening during the spray drying process. So for example, here atomization, really the key thing there is defining your target particle size. And there's of course a variety of different atomizer types and geometries available to finely tune that as needed for a given product. The drying conditions shown here, kind of the core part of the spray drying process of basically drying the droplet into a particle. As mentioned earlier, there's a lot of the tuning handles here can really define the product morphology, the water content, or the physical state. And just as an example, in the cartoon in the lower left-hand corner, linking kind of the formulation and process, here we're showing a problem statement we see occasionally of trying to purposely impede the protein from the surface of the droplet and ensuing particle due to stability reasons. So here we have a surface active excipient, so a formulation decision, and linked to, in this case, we might purposely dry under colder, wetter, slower conditions to allow the surface active excipient to have higher mobility for a longer period of time and migrate its way to the surface for these stability reasons. And additionally on the right, it's the collection efficiency, which often gets overlooked. So a lot of these biotherapeutics are very high-value products, and the geometry of the cyclone can impact the collection efficiencies and whatnot. So for a 2-micron particle for inhalation, you might use a different geometry as opposed to a 30-micron particle for intranasal or reconstitution purposes. Slide 11 just shows some of the offline tools we've developed to define the inputs into the spray drying process. So we'll run through a couple of these, but the upper left-hand corner is the nozzle test stand, so it uses a phase Doppler particle analyzer that we can put different atomizer geometries and conditions offline outside the spray jar, characterize the droplet sizes, and basically match droplet size distributions across spray dryer scales, or maybe for other optimization of particle size, not scale related. Similarly, there's, in terms of the inputs into the drying process, we can use kind of this thermal temperature gradient hot bench experiment and define key thermal limitations that can be inputted into the inlet and outlet temperature constraints of the dryer. As Jeff mentioned, water content, we can link water uptake versus relative humidity with dynamic vapor absorption experiments for a given formulation, link that to the thermodynamic operating space where we kind of can define what the outlet RH of the spray dryer is, control water content in that manner. And likewise, we can utilize CFD models to optimize the drying chamber geometry as well as the cyclone geometry to hopefully meet our desired product profile with minimal compound requirements and development times. So slide 12 just gives an overview of kind of how we use some of our thermodynamic process maps and some of the inputs that might go into them. And this is more of a general example. But on the left-hand side is kind of a process map build. And on the y-axis you see the specific drying ratio, which is simply just the bulk liquid to bulk gas ratios. Can be scalable across multiple or any spray dryer scale essentially. On the x-axis is the inlet temperature of the spray dryer, and using the model on the right, knowing that most of the spray dryer is indeed in the outlet temperature, we can use simple mass and energy balances around that control volume to draw these outlet temperature contours seen by the different colors on this map. Additionally, using those same mass and energy balances, you can overlay relative humidity contours on this plot. So some of the constraints we can overlay, might simply just be a process efficiency constraint. So what's the minimum flow rate we'd want to run at with a given dryer scale to where the run times and cost of goods and efficiency make sense, or it could even be a solution stability constraint, identified early on in the process. The second constraint shown in this example follows an outlet temperature contour. So this could just be a thermal stability constraint of a given formulation where if the material is at that outlet temperature for too long of a period, you might start to see degradation. The other constraint shown here follows an inlet temperature contour, which could just be something like formulation browning at the hot spot of the spray dryer, although we try to minimize that, where the drying gas is actually coming into the spray dryer. Both of these can be identified or solved through kind of that offline thermal gradient strip shown in the previous slide. And kind of the final constraint could be a relative humidity constraint. So you see it follows that 10% humidity line there. And this should be if the formulation glass transition temperature is too low such that the particles don't actually dry down to a solid particle and thus have yield issues, or even more simply, it could just be a water content issue that's such that if you're above 10% and the water content is too high in the final dried particle exiting the spray drying unit. So slide 13 provides kind of this flowchart methodology. It really is designed to kind of logically guide formulation and process parameter selection. And it really kind of hinges on a fundamental understanding of the problem statement and compound properties shown in that upper left-hand box, and then goes through these other boxes that utilizing these offline tools to define a suitable process map, with minimal compound needs and development times. So you can see some of these, the hot bench and nozzle test stand experiments feeding into this, final box that hopefully meets all of the required things like formulation integrity, understand the potential storage and packaging needs, and other spray drying specific things like water content and particle size. So at this point, I'll hand it back over to Jeff to go through some of the solid state formulation requirements.
Cool, thanks, Devin. So as Devin was mentioning, what sort of goes hand in hand with the engineering process of the formulation. And so the ultimate goal is to convert an aqueous spray solution using spray drying into a solid state dry powder. And ultimately, we need to characterize those powders, understand what we've created, and to see if we've actually solved our client's problems, and if it's actually an advanceable process and advanceable formulation. And one of the things to do, obviously, is to include into that a rational formulation design. And what's important for that is the first bullet point here, which is a excipient selection. And there could be a range of reasons why someone may or may not include an excipient in the formulation. The first and probably most importantly is protein stabilization. This is there's a couple different reasons we'll touch on here. One, there's a few different theories around excipient stabilization of biotherapeutic, one being the hydrogen bonding theory, one is the high viscosity sort of glass theory, actually containing or mummifying, if you will, the biotherapeutic, and the third is sort of the water shell theory, which actually is a sort of a hydration theory or to maintain the tertiary structure of the biotherapeutic itself. And there's also bulking reasons why someone might want to include an excipient, as well as handling, which gets back to powder filling and downstream handling and application to the patient. And a comment to that is the analytical aspect of our characterization, which is what kind of data are we going to generate, how are we going to use that data, and how will that actually be applied to the program, why is the data valuable? These are actually questions that we routinely ask for ourselves and our client during a program. And what dovetails off of that are both solid state analytics as well as solution state analytics. And the third bullet point there highlights some of our key tools we use internally, which is solid state analytical tools, looking at sort of thermal properties, such as the glass transition temperature. As Devin was mentioning, water content for biotherapeutic is critically important to understand and maybe perhaps modulate. We routinely operate in the very low end of water content, around 2 to 4% of water content by weight. We need to understand what that water content may or may not be doing to the biotherapeutic itself. We need to understand how the excipients actually organize or not organize in the solid state, and X-ray diffraction helps with that. And we routinely just want to see our particles, and the SEM will do that as well. Now, what's also handy or nice about biotherapeutics is there's a whole tool set around solution state analytics, which are available internally and externally with our collaborators and clients. And tools such as circular dichroism, or CD, is important for understanding secondary structure. We also have internally STS Page, SEC mols, and our clients, we do not often outsource solution DSC studies to look at actually stability of the protein after resuspension of the dry powder. And as I was mentioning, there are a number of reasons why someone might want to formulate a protein. One, as Devin was mentioning in an earlier slide, is simply to protect the API from the spray dry process. But more importantly, what we routinely see is formulation to stabilize the API or the biotherapeutic in the solid state during downstream handling and storage, and also application. So clients have come to us and asked us to look into neat formulations, that being no excipients. Now, included routinely here are excipients such as buffers, such as citrates, maybe some salts, some phosphates. Things of that nature. We can spray dry these, and we have seen really stable formulations using no excipient sort of approaches. But what we lack there with the no excipient approach is a stabilization aspect to that formulation. Now, we have included excipients for stabilization, as we mentioned, bulking of the dry powders, handling, as well as performance. Performance is critically important for inhalation delivery. We need to have an aerosolizable dry powder, not just one that we can produce and stabilize, but also will aerosolize with a high fine particle fraction during delivery. These are all reasons to use perhaps excipients. Of course, we want to mitigate the need or use of excipients for downstream, obviously, don't impact any tox aspects of their burden in the formulation. On this slide, we have, as we've been mentioning, a number of reasons why someone may or may not use an excipient. The first few reasons are bulking, API stabilization from engineering state to state, engineering process, also solid state. We also need to stabilize the particles themselves. We often use non-hygroscopic high TG excipients to limit mobility of the formulation of the molecules in the formulation in relation to each other, also in relation to the API itself, which then would obviously create a long-term stable formulation. We also want to mitigate particle agglomeration, reduce sticking upon storage. This is also important upon for its versatility and aerosolization. So we might include surfactants. These might be, say, a low level of tween in formulation, or other sort of non-hygroscopic amino acids. This is a high-level overview of a toolkit that we have internally at Bend Research. We are often asked what sort of tools we actually can apply to programs. This is to give all the listeners sort of a rough glance at what we actually can apply from the solid state aspect as well as the wet lab aspects. And it's not necessarily the toolkit that's important, but how you use the toolkit. And so it's often a discussion point upon the first few meetings with the client as far as what sort of data set we want to generate in the solid state and the wets, and also the wet lab state, to actually define a process and a formulation that's advanceable to the GMP, and also the clinic. What's important that comes out of all the analytics is really a guidance map. From the engineering standpoint, we develop guidance maps to understand how our process impacts the product. And the downstream angle, from the formulation characterization aspect, what we're doing, we want to develop guidance maps to understand what's actually happening in the solid state, if we've actually achieved our goal of creating a stable formulation, and how that stable formulation might be impacted negatively or positively in the face of, say, solvents, or in this case, water. What we routinely do is a glass transition temperature analysis as a function of relative humidity in the environment to understand tolerance of the formulation in the face of RH. Along with that, we'll often perform vapor sorption assays where we actually look at water uptake in our formulations, understand how that impacts the properties of the excipients. In this case, you can see the sugar, or trehalose in this instance, crystallized out at around 50% RH. So these are important guidance maps we often talk about with our clients, so they can understand downstream handling and processing of their powders so they don't impact negatively the performance and application. These sort of concepts jump into our first case study, which we'll dive into now. There are 3 case studies, the first one being a collaboration with our partners PATH and Fraunhofer. This was a 2-year Defense Threat Reduction Agency funded program where we formulated and developed a stable and scalable formulation of a flu antigen that Fraunhofer was providing in the GMP conditions. This was a neat program. We actually applied solid state characterization, sprayer expertise, and also solution state applications. Our clients did some analytics that sort of justified and proved our concepts. So as I mentioned, the goal of this program was asked to us by DTRA was to actually develop a formulation that was stable, room temperature stable, could be exposed to elevated temperatures, say out in developing countries. And the biopharmaceutical we were using was a HAC1 flu subunit antigen, which was produced under GMP conditions. It was a well-behaved API, and we applied, of course, spray drying to develop this program. What we initially did for this program was sort of routine sometimes. It was sort of an empirical, rational approach to formulation decisions and development. We screened a number of formulations containing different ratios of excipients, and then expose them to our toolbox, or run through our toolbox. The first one of our first order tools we use is DSC to actually develop a glass resistant temperature readout to understand what the TG looks like in the dry state, which is important from a mobility standpoint and temperature tolerance standpoint of these formulations. It also tells us if we're seeing phase separation, if we are in fact getting amorphous dispersion of our particles during manufacturing, or if we're getting things that are separating unstable and generally poorly behaved. And this was a dry TG analysis we did of a number of formulations as shown on the screen. Our sort of minimum baseline, if you will, for our TG, which was a go, no-go, was around 75 degrees Celsius. We saw that certain excipients, not surprisingly, the larger excipients actually bumped up our TG. These were actually dispersions of salts, sugars, and polymers we used here to actually develop a rational sort of approach for formulation development decisions. Using this tool, the DSC, as well as things such as PXRD, SEM, we actually then pushed forward and developed a protein formulation for a stability study, as shown here in this slide, We performed SEM across 6 months at a number of conditions from the stability study at negative 25, 40 degrees Celsius, 50 degrees Celsius, and saw stability by SEM. We also saw stability by DSC. We produced 3 lots, different conditions, and saw that the TG, this lead formulation, was held strong at 120 degrees Celsius, which is great, did not phase separate. Our collaborators, Path and Fraunhofer, did some in vitro analysis looking at potency across 6 months, 50 degrees Celsius and 40 degrees Celsius of this HAC1 antigen, and actually saw that in this formulation of these conditions, the potency was maintained and held strong, which was great from a in vitro standpoint, but obviously to prove this in vivo, we also, at Fraunhofer, they produced a stability study, or analyzed the stability study, powders, using an in vivo challenge, and looked at antibody titer as a function of inoculation dose after the powders are stored either at -20, 4, or 40 or 50 degrees Celsius. And as we can see, compared to the control, the HAC1 on the far right, our formulated powders actually maintain the potency as the controls show across a 6-month as well as 3-month stability study. So overall, this is a successful program, which is actually currently wrapping up and might be ongoing, but mostly wrapping up currently. Manuscript is in the works. The second program is an antibody program we worked on with our collaborators at Pfizer. The goal here is to develop a formulation around high concentration reconstitution of a dry powder containing monoclonal antibodies, an IgG. Obviously, the formulation handles here in this case where we needed a spray dry solution that was actually going to stabilize the protein. We needed to modify the process handles during spray drying and then characterize the final product. Obviously, we want the good protein, not the bad protein in this case. And what we actually as defined was we wanted the dry powder. We could resuspend quickly. We needed to optimize the process space using SEC, and as Devin mentioned previously, using the sort of operating space guidelines, we developed a spray dry area that maintained the quality of the powders looking at T in and T out. We also used SEC to look at the quality of the API, or in this case the mAb, coming out of the formulation after resuspension. And the team also performed a 3-month stability study at 5 degrees Celsius, 25 and 40, and saw that we did see some aggregation form over time, but mostly we were able to mitigate the aggregation and produce a high-quality powder using a rational spray-dry process and formulation. So this orange dot in this operating space, for spray-dry operating space Devin touched on earlier, the orange dot on the right-hand side of this plot, sort of the standard space that most sort of, say, lab-scale Buki spray dryers operate at. We used a modified PSD1 spray dryer to sort of make the inlet or drop the inlet temperature to actually make this more gentle process for the monoclonal antibody to actually mitigate aggregation. What we found was that it had a significant impact on mAb quality after resuspension. In these 3 pictures here in the middle of the slide, we see on the far right sort of cloudy solution, which are insoluble aggregates of mAb. In the middle image there is the water reference, and the far left is our improved condition spray dry process, which led to a improved clarity and middle aggregation, sort of justifying our process map and a rational spray dry process decision tree that we developed for this program. This slide highlights the temperature conditions that were used during that spray dry process. We ranged from T in of 120 to 160 and the T out of 60 to 83, as shown that in that middle sort of plot there, spreadsheet there. This all, as Devin was discussing earlier, water content sort of correlates with formulation as well as T in and T out conditions. And one of the conditions we also were looking at was downstream handling, so here is flow. The higher conditions actually were causing a plasticization of powders and actually impacting sort of what we call the stickiness of those powders and handling. We actually looked at also using SEC API integrity of these powders, of the biotherapeutic IgG of these powders, using either a initial cooler temp or a hotter temp condition or a modified PSD1. Compared to the reference, we see we actually decreased a large aggregate formation that was occurring using the hotter temp conditions that were brought to us. So the process did, in fact, mitigate aggregation as shown by SEC as well as the photograph we saw earlier. We also looked at API quality, IG quality is here using FEC as well as SDS page. In this formulation, we're using sugars such as trehalose. We're also using amino acids, phosphorus amino acids like histidine. We're also looking at surfactants, such as tween 80, polysorbate 80. These tween conditions were 0% by weight tween, 1.47% tween 80. And these were this was a proof of concept study. We realized these tween conditions were fairly high. This was simply a proof of study, knee-jerk program to look at, can we do this? Can we actually modify mAb quality using formulation and process? And as we've shown, we actually saw an increased resuspension rate with the higher tween contents and a decrease in precipitates using surfactants like tween. And since this program, we've actually been looking at lower levels of tween to actually impact particle quality and resuspension rates themselves. It's an ongoing effort. And that brings us into our third case study, which Devin will be touching on, which is one of the programs he worked on, which is the formulation manufacture of a spray-dried norovirus vaccine for intranasal delivery.
Thanks, Joe. So slide 34 just provides some background on the norovirus, which we won't pretend to be experts on. The collaborator in this case was the norovirus expert. But essentially this is a fairly common ailment that probably a lot of people have experienced, unfortunately, at some point in their life. Very common in hospital settings, schools, cruise ships, and other military branches where people are in highly confined spaces for longer periods of time. The molecule we were working with was a non-enveloped virus-like particle. And of course, there's some other information around the actual norovirus available here. On slide 35 kind of provides the high-level problem statement of developing a spray drying process using a lyophilized solid formulation for intranasal delivery. There was data available to support smaller particles had higher efficacy, but there was concern on the extreme small end of having the particles be introduced into the lungs. So kind of one of the primary particle size goals was minimizing particles in the inhalable range, shown by that D10 value being, or target being greater than 10 microns. Of course, there's some other spray drying constraints listed as well, but the focus of this is more on the particle size, particle engineering aspect of it. So, with the particle size being important, atomization was obviously one of the key challenges or focal points of the program. So, given the scales and equipment, spray drying equipment we were using, this table just kind of shows some of the different atomizers, so whether a two-fluid pressure swirl or ultrasonic nozzle, and just kind of some of the advantages and disadvantages commonly known throughout literature of these different atomization techniques. Slide 37 shows some of the actual particle distributions produced with these different nozzles. So with the ultrasonic, you can see a very tight span, which is well known to these ultrasonic nozzles, but the particle size, there wasn't enough tunability in that energy inputs for that atomization method to increase the particle size much higher than what's shown. Similarly, the two-fluid nozzle, though very, very highly tunable nozzles, typically are naturally produce a lot of fines seen by that kind of tail on the sub-10 micron range. Whereas with the pressure swirl atomizer, we were able to meet all of the particle size needs as well as the span requirements for the right delivery particle size distribution. With this particular formulation being a very viscous solution in low solids, we had to produce rather large droplets that dried a little bit maybe more slower than we were used to end up with a dried product out of the spray dryer. So we adapted our equipment with these spray drying extensions to basically increase the residence time or drying time, which was required to achieve of course the right particle size, but also some of the other metrics, namely water content and yield, and especially at the smaller batch sizes we were working with. Slide 39 just shows kind of a comparison of the lyophilized milled product and the spray dried powder and the morphology and kind of particle sizes produced from the two methods. And slide 40 shows the immune response in rabbits there's actually 2 virus-like particles in the formulation. So you can see the comparison of the lyophilized and the spray-dried material at a couple different doses, but both gave comparative immune responses, suggesting that the spray-dried material remained in its efficacious state throughout the spray drying process.
So I guess to summarize this work overall, with a broad brush here, we work with partners, collaborators to spray dry manufacture biotherapeutics and develop these robust methods from formulation standpoint as well as an engineering standpoint. And we hope to describe in this webinar sort of a high-level approach to developing guidance maps from an engineering standpoint as well as from a formulation standpoint, which touches on understanding of both processes and then problem solving, and the overlay of that being in analytics and the application of analytics to solve these sort of problems. Ongoing work isn't actually happening internally to look at vaccine for inhalation delivery and other client work as well. So we appreciate you listening to our webinar today. If you have any questions, please feel free to get in touch with us. And we thank you very much.
Great. Thank you to both our speakers for that insightful presentation. Why don't we go ahead and start that Q&A session that's part of today's webinar. So at this point, I just want to point out that we have displayed here Bend Research's contact information, so be sure to jot that down in case further questions arise. Also, be sure to continue sending in your questions and comments using the questions window. Now, I've already received some questions, so I'll go ahead and start with those. So our first question to kick off our Q&A portion of this webinar, what is the high temperature which the biologic will see at the inlet? So could our speakers tell us a bit more about those temperatures?
Yeah, that's a great question, actually. Devin, you want to touch on the inlet-outlet temp and how we bracket sort of those conditions with a high TG material?
Yeah, so a lot of the gas dispersers, are in the spray dryers are designed to have very kind of intimate mixing with the drying gas and atomized droplets, such that while there is drying gas, at said inlet temperature, surrounding these droplets, this evaporative cooling process that's very immediate actually kind of shields the product from the inlet temperatures. That being said, there is some recirculation in the spray jar, so you can have particles kind of reintroduced into that gas stream. So that's where we use some of the hot plate experiments or maybe other thermal degradation analysis to kind of define what that maximum inlet temperature can be, especially if we're talking about longer processing times and whatnot.
And what's important there as well is developing a high TG formulation that actually can tolerate the outlet temperature, which is actually usually the highest temperature observed by biotherapeutic. We usually rationally develop these formulations with small molecular weight sugars as well as, say, a polymer, for instance, with biotherapeutics, to actually develop a higher TG material so that we don't see mobility at, say, a 60-degree outlet during a jar collection, for instance. So the biotherapeutics themselves often have TGs in the dry state around 190, 200 degrees Celsius. So we really are seeing a stable product and formulation using these conditions during collection using spray drying.
Great. Thank you, both of you, for addressing that question. Our next question comes from an attendee who writes, can you explain the DVS experiment in more detail? In brackets, this person has noted questions such as, what material are you evaluating in the DVS instrument? Is it the dried material? So again, I wonder what the two of you have to say about that.
Okay, great. That's a really good question. So the typical process might be, doing a quick formulation screen on our small scale spray dryer and actually producing very small quantities of dried powder for a given formulation, and taking kind of our favorite powder from that so it has been spray dried and aliquoting a certain portion of that to the DVS experiment. So basically we are looking at the water uptake of the dried powder as a function of equilibrium RH, and relating that back to what the spray jar outlet RH is, and can relate that to an expected water content. Gets a little bit more complicated in the fact that, as we mentioned, the residence time in the spray jar is not very long, and so whether that particle actually reaches equilibrium is another issue. But we've developed some unique solutions to deal with that for when we need really precise water content control of basically surrounding the collection container in a controlled temperature environment, such that we can ensure the exact RH, uniform throughout the entire collection process. So it's almost like an in situ equilibration step, when precise water content control is required.
Great. Thanks again for answering that. Our next question, does hydrogen bonding between API and excipients necessarily ensure stabilization?
No, that's a good question. Yeah, I think from the standpoint of proteins, it's an important hypothesis around why people use sugars. We try to combine that theory along with the high TG sort of theory, looking at polymers in matrix, amorphous matrices, that actually stabilizes that. And so we use we push a high TG formulation approach over the hydrogen bonding approach. We do use those hand in hand to actually understand how formulation will stabilize a biotherapeutic. As I mentioned earlier, there are a few different theories out there around hydrogen bonding and excipients and protein stabilization. So we try to combine that mode of thinking along with the classic amorphous dispersion high TG approach for some molecule formulation as well.
So essentially it can be like a blend of thermodynamic stability versus kinetic stability.
Exactly. Thanks, Devin.
Great. Thank you for to the two of you again for collaborating on that response. Our next question, does the hot drying gas lead to protein degradation during the spray dry process?
So yeah, that's very akin to the first question, and I guess the short answer is hopefully if we've done our homework right, no. But if some of the offline tools that we've developed are designed to where we don't have to go and do several spray drying experiments of pushing up your inlet temperature or down your inlet temperature until you see failure. So really designed to know what that temperature is offline and process at a range where there won't be protein degradation issues.
Great. Our next question, what are the possibilities of using a solvent mixture or an organic solvent for particle engineering of biotherapeutics using spray drying?
Yeah. Sorry, then go ahead.
Yeah, so a lot of our spray dryers we routinely process organic solvents on. In particular for the biotherapeutics, it's it's a balance of you kind of utilizing the same tools we discussed. You can manipulate your process around, instead of water content of an organic solvent, concentration on the outlet material. That's also to be balanced by acceptable PDE limits and things like that. But it's not uncommon for us to have small to moderate amounts of organic solvents in these solutions. All of our spray jars operate with nitrogen drying gas, so it's an inert atmosphere, and we've developed the solvent handling programs for dealing with flammable solvents.
And I'll jump in there too. So there's obviously has to be a need to use an organic with a biotherapeutic, and we can see those sort of needs with solubility issue potentially. We do have GC headspace sort of analysis in-house to actually monitor if we are going to use an organic solvent, what that actually looks like from the powder standpoint as well. So we have used those systems Tim was mentioning. We can use those again, but we do try to shy away from using organic with biotherapeutic unless there's an obvious driver process or solubility issue driver there that pushes us in that direction. And if we do end up going that direction, we can watch that over time and actually regulate those concentrations.
Yeah, and just for the case study 3, for example, it was a combination of an aqueous organic solvent blend.
Great. Thanks again for both collaborating and putting your input into that question. So our next query comes from another attendee who writes, how about using amino acids as surfactants?
Yeah, that's actually a good question. So amino acids can be used as stabilizing excipients. We've seen that with arginine and histidine. We've also seen those used as surfactants, actually very popular in the inhalation field, such as leucine. So we do actually have applications for amino acids in both areas from a formulation standpoint, one being API stabilization, the other being particle sort of flowability or particle handling modulation, and also to compete off the API for the surface potential as well. But that's an interesting conversation point actually for a broader discussion, but that's something we've looked into and continue to look into.
Great. Our next question is about excipients. What excipients are often used for formulation?
Yeah, I actually had a slide here I threw out right before the talk, but basically there's a laundry list of excipients that are used for biotherapeutic formulation. What we balance that against is performance and toxicity. Obviously, for certain applications, you can or cannot include certain excipients. We routinely see and utilize buffers, molecular weight buffers like citrates. We see salts, phosphates. We do see different excipients used in spray drying than we do in lyophilization. So there is a rational sort of approach formulation that is different for spray drying and lyophilization. For instance, we would try to use excipients like mannitol or trehalose as opposed to sucrose for performance issues. And we do often use high molecular weight, sort of higher TG excipients that act as surfactants, like a skinning polymer, like a PVP, to actually modulate the surface functionality of these particles, as well as to perhaps compete off the surface of the API from these particles as well. So there's various reasons to use excipients. The list is fairly long, but ultimately we want to decrease the burden of the excipient in a formulation so that we use only what's necessary. So we actually will only use those that are sort of a value-added solution to a client's problem around formulation and spray drying.
Great, and it looks like we're coming up close to the end of the hour. So it looks like we have time for one more question, and that question is, how about using acoustically levitated droplet drying characterization on formulation and process development by spray drying?
Yeah, that's a good question. There's obviously a lot of literature around of people using that acoustic levitation device and tracking, temperature profiles in the spray dried material, as well as even, collecting that single particle and doing some solid state characterization. We developed similar tools, whether it's just a small scale manufacture on the mini scale spray dryer, or we've actually taken that same approach and inserted a droplet into a drying environment, so a given temperature and relative humidity environment, and put that droplet onto a tip of a very small thermocouple where we can actually track the drying profile, so we can watch the temperature approach the wet bulb temperature of the given processing solvent, and then increase back up to the outlet temperature, as well as some high resolution imaging, similar to how people use the acoustic levitation device. So it's a really cool device. It's been developed, and it has a lot of advantages. We found some other ways to draw a lot of that same information out of kind of what's going on at the individual particle, from whether a temperature profile, drying morphology perspective, or even some of the solid state characterization of those droplets to particles.
Great. At this point, I'd like to thank both of our speakers for delivering such a great presentation, and also taking the time to talk us through all those questions that we got in today. So I'd also like to thank all our attendees for participating in today's conference and for submitting all those great questions to us. We hope that everyone has found this conference informative and entertaining, and we hope that you have a great day.